everyone, how are you all? Hope you are fine. Welcome to Rudy's Edu Vlog. Today we are about to see some interesting information about genetic engineering. Without any further ado, let's get into our video. The field of modern biotechnology started when recombinant human insulin produced by bacteria was first marketed in the United States in 1982. The effort leading up to this landmark event began in early 1970s when research scientists developed protocols to construct new types of bacterial plasmids or vectors by cutting out and pasting pieces of DNA together to create a new piece of DNA called recombinant DNA that could be inserted into the host bacterium like E. coli. We have also observed that yeast can be made to produce vaccines like hepatitis B, plants having special properties like resistance to certain diseases, pests, and herbicides, and plants with superior nutritive qualities can be generated very efficiently. These excellent goals of genetic engineering were achieved because of the advent of recombinant DNA technology. Recombinant DNA technology is one of the few techniques that made the conventional biotechnology into modern biotechnology. Paul Berg, Herbert Boyer, Annie Change, and Stanley Cohen are the team of scientists who made the first recombinant DNA molecule in 1973. Simply defined, it is the art of cutting and pasting genes. There are, however, many new applications of this technology invented each year, and it is impossible for any textbook to be completely up to date. This technique encompasses a number of methodologies or tools that enables to construct new combinations of DNA, recombinant DNA or rDNA, in the laboratory for different purposes. The rDNA molecule thus constructed can be introduced into an appropriate host cell where they can be multiplied and generate many copies. This forms the basic concept of the processes known as gene cloning or DNA cloning. The technique of gene cloning can generate unlimited copies of a DNA molecule, example recombinant DNA, by replication in a host cell, and it was first developed in 1970. The following are some of the major applications of rDNA technique: genetic mapping, DNA sequencing, mutation studies, transformation, genetic engineering, recombinant DNA libraries, restriction enzyme sites analysis, analysis of gene transcriptions, polymerase chain reaction, PCR. The goal of rDNA technique is gene cloning to generate large amounts of pure DNA that can be manipulated and studied. The following are the basic steps involved in the process of rDNA technique for gene cloning. Step 1. Isolate DNA from organism, that is extraction. Step 2. Cut DNA with restriction enzymes to generate DNA fragments of desired size, which may have a specific type of DNA sequence or gene that has to be cloned. Step 3. Ligate or splice each piece of DNA into a cloning vector to create a recombinant DNA molecule. A cloning vector is an artificial DNA molecule capable of replicating in a host organism, example bacteria. Step 4. Transform recombinant DNA, that is the cloning vector plus DNA fragment, into a host that will replicate and transfer copies of progeny. That's it, my friends. Hope this video is helpful. Hope you like it. So do like, share, and subscribe. Comment below if you need any more topics to be covered in recombinant DNA technology. Thank you, everyone. Have a great day. Happy New Year.